Welcome everybody to today's COSI seminar. Today's speaker is Professor Smalyukh from the Department of Physics and the Liquid Crystal Material Research Center here at the University of Colorado. Professor Smalyukh holds not one but two PhDs, one from Kent State University on chemical physics, the other one from the Supreme Attestation Commission of Ukraine on optics and laser physics. He has produced more than 40 refereed publications, chapters in books, to international conferences on the field of optics and liquid crystals, and gave numerous invited and plenary talks at different conferences. His contribution also resulted in recognition by the International Liquid Crystal Society, who granted him the Glenn H. Brown Prize from that society. And today he will be talking about non-contact optical control of multiple defects and structures in liquid crystals using holographic and timeshared optical trapping. So let's welcome Professor Smalyukh.
Okay. All right, thank you very much for this opportunity to speak here. And since I know that your IGERT program is focused on imaging and optics, and therefore today in my talk I'll focus more on the optics and imaging and optical manipulation parts of the research in my group. And so what you are looking at here is some sort of a preview of my presentation. So those movies demonstrate how we can use tightly focused laser beams for manipulation of colloidal particles in 3 dimensions using holographic optical trapping approach, and also how we can use those tightly focused laser beams to manipulate structures in liquid crystals. And so I would like to start this presentation by highlighting the iCamp Summer School, which we organized in China this year. It will take place in 4 different cities in China. So 1 week in Shanghai, Hangzhou, 1 week in Qingdao, and 1 week in Beijing. And the focus topics are all optics related. Materials for optics, it's Intercontinental Advanced Materials for Photonic Summer School. I hope that students, postdocs here can apply for the fellowships that we can provide up to $2,500 to participate in this summer school. And so this will be a combination of advanced education and also, tourism, sightseeing, learning about diverse cultures. So the focus of my presentation today will be optical manipulation of different structures, defects, and particles in liquid crystals. And again, what you see in here all is done by tightly focused laser beams. In other words, by light, we can manipulate matter, and you will see later on that we can also use matter, liquid crystals in particular, to manipulate properties of light too. And so I'd like to start with this a very brief introduction into how optical trapping works. And so if you have a tightly focused laser beam, as shown in here using a high numerical aperture lens, then you realize if you have some colloidal sphere that has a refractive index different than that of surrounding medium, because of refraction at those surfaces of the sphere will deflect the laser beam into a different direction, right? And so in a sense, you pushed the photons in one direction, the photons push on the particle in the opposite direction, and so therefore there is a restoring force which will push the particle to the center of this beam. Now the same is also true if you consider the direction along the optical axis of this tightly focused laser beam. Again, because the sphere is changing the direction of light propagation, it's changing the direction of photons in which they are moving, the photons again exert the force in the opposite direction, and eventually the particle will be trapped in the center of the tightly focused laser beam. We can think about the forces which can be produced by those tightly focused laser beams in terms of using simply dimensional analysis. And so this force will be nothing else but power divided by the speed of light. And this gives us a fairly good estimate for what kind of forces we can produce using laser beams. And those would be forces of the order of a piconewton per milliwatt of laser power. Of course, those are relatively small forces, but for the micron-sized objects, those are actually huge forces which are much bigger than, for example, the gravity forces, which are 3 orders of magnitude smaller for micron-sized particles usually. And so we can also think about laser trapping in terms of the scattering forces and gradient forces. And so the scattering forces result from simply backscattering of light, right? So the photons bounce from the interface of particle with the surrounding medium, and those forces are proportional to intensity of the laser light, and they also act in the direction of light propagation, right? But then there are also the so-called gradient forces, which result due to strongly non-homogeneous distribution of light intensity near to the focus of the laser beam, as you can see in here. And those forces are proportional to the gradient of laser intensity. When we have the gradient forces larger than the scattering forces, the particle can be actually trapped in the center of the tightly focused laser beam. For this, one of the important conditions is that the refractive index of the particle is larger than that of the surrounding medium. And so, of course, it's good to be able to trap a single particle, but for many experiments, exciting experiments, we need to trap multiple particles, and there are different approaches that would allow us to do this. One of those approaches is based on the spatial light modulator, shown in here, which allows us to control laser intensity distribution in the focal plane of the objective, right? The other approach is based on different ways of steering the laser beam. So we can steer the laser beam in the focal plane of the objective using, for example, galvomirrors or using acousto-optic deflectors. And because we are able to steer this beam relatively fast, we therefore would be able to manipulate multiple particles by a single beam which is moving very fast, so that we can manipulate multiple particles. And so the advantage of the holographic optical trapping approach, which you can see in here, as compared to the steering-based approaches, is that not only we are able to produce the arrays of optical traps in the focal plane of the objective, but also it's possible to manipulate particles below and above the focal plane. So in other words, it's possible to manipulate those particles in 3 dimensions. Moreover, there is another advantage that the spatial light modulators, such as liquid crystal, SLM, allow us to do, is that we can generate different types of laser beams, including, for example, LG beams of different types that you can see in here. And later on, you will see how those can be useful for manipulation of laser trapping once again. And so how this holographic optical trapping works. Well, imagine you would want to produce multiple focused laser beams in the focal plane of the objective. This is one way you could do it, right? You could simply have multiple beams which could be focused in the focal plane of the objective by using this telescope and the objective. But there is another way to do it using simply a diffraction grating, and the very simple demonstration of this holographic optical trapping approach was done actually with microfabricated face mask in silica that you can see in here with some periodic structure that would then produce an array of focused laser beams in here in the focal plane of the focused laser beam, and this would allow one to trap multiple particles. Of course, in our approach, we use liquid crystal spatial light modulator, which you can see in here, and this is the scheme of the optical trapping system in our lab. Basically, we have two telescopes in here, right? And we have liquid crystal spatial light modulator, and we also have some optics to control polarization of the laser beam. This is the actual photo of the system, how it looks on the optical table in our lab. And this is spatial light modulator. The fiber laser brings the laser beam in here, and so we have two telescopes, one before spatial light modulator, one after, and then the laser beam is brought into the so-called laser port B on the right side of the microscope. But in our research, as you will see, we'll also need to do imaging in 3 dimensions, not only just trapping and manipulation, and this we do with the fluorescence confocal polarizing microscopy, and the scanning beams which we use for imaging in 3 dimensions are brought in here from the left side port, right? So the left side port is used for imaging, the right side port is used for trapping, and then there is also a fast camera you can see in here, which is also used for imaging, but in regular transmission microscopy mode. And so in here you can see what we can do in terms of manipulation of the laser beam, right? We can produce all kinds of laser traps with different shapes, periodic patterns of laser intensity in the focal plane or in 3 dimensions, not only just in the focal plane of the objective. And so one example what you can do with this kind of system is shown in here. This is biology-related research when we trap arrays of biological cells, bacteria which are alive, being trapped by those tightly focused infrared laser beams. So we can do different types of research projects related to biology, for example, and laser trapping and manipulation has been extensively used in biology. But today, my research, as you will see, will be mostly focused on soft matter applications of optical trapping and imaging in 3 dimensions. And so what you can see in here is a movie how we manipulate particles in 3 dimensions. So you can see some of those particles being in focus, the other particles are out of focus, and so the reason for that is that we move them separately at the same time in 3 dimensions, right? Not only just in the plane of the image, but also we can push them upward or downward with respect to the focal plane of the objective. And then here you can see the calibrated positions of those particles. So those positions denote how far from the focal plane of the objective the particle is actually trapped. So you can see that some of them are out of focus and some of them are in focus, those central ones. Again, in here, those are just some snapshots from this movie showing us where in the 3-dimensional space of this sample the particles were actually located, right, with respect to the focal plane of the objective. Now, as I mentioned it, most of the applications applications I'll show today will be related to liquid crystals. And this slide is to show you how we distinguish liquid crystals, anisotropic fluids, from both solid crystals and isotropic fluids, right? So liquid crystals are unique states of matter which combine properties of both solid crystals because they have some order, orientational order, and also isotropic fluids because they can move, they can flow, and the molecules in here are not restricted in their position, so they can change their position with time. So therefore, this material can flow. And now we will need to do three-dimensional imaging of molecular orientation patterns in those materials, right? So you probably know about fluorescence confocal microscopy, which is widely used technique in different branches of science in order to image three-dimensional positions of different objects in samples, right? For example, you can use it to image biological cells. When you will label different components of this cell with different dyes, you can get an idea about structure of this cell. Now, in liquid crystals, we can have one compound, one chemical compound forming this liquid crystal material, and the only thing we would care about it is the orientation of molecules, right? Because everywhere in this space we could have the same material, right? But orientation of those molecules will change in this three-dimensional space. And so how can we image three-dimensional patterns of molecular orientations in those liquid crystal samples? Well, all we do here is we slightly modify fluorescence confocal microscopy by introducing a polarization rotator so that we can excite and detect fluorescent light in polarized mode, right? And so now, if we also use dye molecules that have their transition dipoles along the long elongated shape of the molecule, such as this one in here, then the fluorescence signal will depend on orientation of those dye molecules. And the dye molecules, because of their elongated shape, follow orientations of liquid crystal molecules. So this means by learning how the dye molecules are oriented in the sample, when we simply dissolve them in very tiny quantities of 0.01% or so, we can this way learn about orientations of liquid crystal molecules. And so in here I would like to show you how we can use this three-dimensional imaging in fluorescence confocal microscopy to visualize positions of particles and also at the same time to visualize structures of liquid crystal. And so what you saw in here on this image is two particles which are trapped at different locations along the depth of the sample. And you can see one of them is in focus, but the other one is not, is far away from the focus at some 25 microns distance along z direction. Now, with the confocal microscopy in polarized mode, we can actually very directly take the side view of the sample and visualize where those particles are located with respect to the structures in this liquid crystal medium. So what you can see in here is the layered structure of the liquid crystal cholesteric material, and the molecular orientations change along those layers in here. Even though everywhere you have the very same type of molecules, but their orientation is changing. So there is a twist as you would go from top to bottom, and you can see where those particles are precisely located along the depths of the liquid crystal sample, right? And so it shows also us that we can manipulate those particles in 3 dimensions in this liquid crystal medium. But not only can we manipulate those particles, we can also use them as handles to manipulate defects in 3 dimensions. And you can see in here how we manipulate this particle that is attached to a defect line. With the confocal microscopy, we can visualize that this line actually corresponds to a dislocation where we have one layer inserted, and with that, with this particle, we can not only manipulate the position of this defect line in 2 dimensions in the plane of the sample, but we can also produce kinks and anti-kinks of this edge dislocation so that we will push the defect line across the layers, right? Across the depths of the layers. So this is just one example of manipulation, but a lot of other exciting ways of manipulation could be done, and you can see those in here. How those are useful? Why do we need to do something like that? Well, first of all, we can learn a lot about properties of defects in liquid crystals, but by manipulating them this way. For example, what you can see in here is a defect line in liquid crystal. By using this particle, manipulating it, we can break this defect apart, right? And therefore, the other side of this defect line will disappear. What this tells us is that this defect line is topologically unstable, right? What that means is the total topological charge of this cluster of defect lines, as we can see now here by doing imaging in the side view of this cluster in the layered system, we see that the total topological charge is 0 because we have 2 lambda minus 1/2 disclinations and 2 lambda plus 1/2 disclinations, as you can see in here. So moreover, we can now manipulate this defect cluster simply from the balance of optical trapping force and line tension. We can determine the line tension of this so-called lemon cluster being 53 piconewton or so. But what I will show you now is actually something that is very specific from the point of view of optical manipulation, and that's very unique for liquid crystal media and other anisotropic fluids. So as you might know, the liquid crystals are birefringent media, right? So for the uniaxial liquid crystal, the effective refractive index will depend on the direction of light propagation with respect to the optical axis of this material, and also this index will depend on the polarization of the used light for trapping. And so the value of this index can change from the so-called ordinary value of refractive index to extraordinary. And so therefore, not only can we manipulate those defect lines by using particles as handles, we can also manipulate them directly by tightly focused laser beams if we use a linearly polarized laser light, as is shown in here. Because in this case, if we consider the structure of this cluster of defects that we can have in here, we realize that for light polarized in this direction or vertical in here, because this is a cross-section, the effective refractive index in the central part will be close to extraordinary value, whereas it will be lower farther away from the center of this cluster. And so we can also see that if we introduce colloidal particles into the liquid crystal medium, their properties of laser manipulation will be also very different than what you observe in the isotropic fluids. And so in liquid crystals, when we introduce such particles, we can treat them, for example, using a surfactant, so that the molecules around this particle will be aligned in some prescribed way, for example, vertically to this surface. And then what we realize is that this particle with molecules aligned vertically to its surface is actually similar to a point defect of this type plus one hedgehog defect that you can see in here. And therefore, if you introduce it into the liquid crystal medium where the far field director or liquid crystal molecules away from this particle are all aligned vertically, then we need to introduce additional point defect so that the total topological charge will be conserved, right? So that because the particle is equivalent to +1 topological charge, we need a point defect of -1 topological charge. And this is what we actually observe in here in computer simulations. And also, this structure is observed in the polarizing microscopy observation between crossed polarizer and analyzer that you can see in here. Now, because this particle is equivalent to +1 point defect and we have -1 point defect, this is a dipolar structure. We can also have a quadrupolar structure where instead of this point defect, we will have a ring of disclination as shown in here, and this will be a -1/2 ring, which is again equivalent to point defect -1. And again, this structure with quadrupolar symmetry of distortions of optical axis is shown in here when observed between crossed polarizers. Now, we can also treat those particles to produce tangential boundary conditions as shown in here, so that in this case we observe simply two bosoms at the opposite poles of this particle. And we can have also a situation when the particle will barely distort this optical axis of liquid crystal medium, right? So all of those situations are possible. But now my question is how this is relevant to optical trapping and manipulation in liquid crystals. Well, let's consider if we have a laser beam, shine it into the liquid crystal medium with polarization which is orthogonal to the far-field optical axis or liquid crystal director far away from this particle. So then, in the color scale shown in here, we have distribution of effective refractive index as shown on this picture, right? So it changes from ordinary value to the extraordinary value, and there is this corona around the particle of refractive index being different than that far away from the particle, right? And then if we consider polarization state which is parallel to the far-field optical axis, then this distribution of laser of the effective refractive index shown again in this color scale is very different, right? So as you can see, by controlling polarization for the very same particle, we can have very different distribution of effective refractive index in this medium. And so we can also have other types of the alignment of liquid crystal molecules around the particles. And again, this corona of effective refractive index will be present in those situations too, but will be very different, will have different symmetry as compared to what you saw before. And so now let's see how we can characterize those optical trapping forces. So very easy way to do it is if you shine the laser beam into one location, trap the particle, and then very quickly move it into the new location and see how the particle is following this change of position of the laser beam. So you can track the motion of this particle very precisely for different laser powers as shown in here. You can take derivative. This will give you the velocity of particle. You can this way find the viscous drag force, and from the balance of the viscous drag force and the optical trapping force, you can reconstruct the trapping force as a function of the distance from the center of this particle. Now, if you do it for two different orientations of polarization, you notice that for one polarization, you obtain the trapping force is stronger than for the other polarization. So the forces of laser trapping are polarization dependent in here, right? As you can see, something that you would not expect and you would not observe in the isotropic fluids, right? So by controlling polarization, you can control laser trapping forces. Moreover, we can also have a situation when the ordinary refractive, the index of particle will be between the ordinary and extraordinary values of the index for the liquid crystal medium. So in this case, we can simply switch linear polarization between two orthogonal states and have the particle being trapped for one polarization state, but being repelled from the particle, from the center of the laser beam for the other polarization state. And this is what you can see in here. The particle is moving back and forth, and you can see that we can trap for this polarization state when polarizer is orthogonal to the far-field optical axis, because the refractive index of particle in this case, in this color scale, is higher than that of surrounding liquid crystal far away from the particle. Now, as we switch polarization in this location to orthogonal, we realize that now we have the opposite situation. The refractive index of particle is lower than that of surrounding medium, which is now extraordinary value, and therefore the particle is now repelled from this tightly focused laser beam. And so we can simply change the duration, the time interval between switching polarization between two orthogonal states and have the particle moving closer and farther away from this tightly focused laser beam as shown in here. Moreover, because in all cases the particle is following the strongest gradient of refractive index in this medium, we can have different particles with different types of corona around them following different trajectories, as you can see in here. So in a way, we can navigate motion of the colloidal particles